All right, today we're going full-on detective. We're diving into a really cool diagnostic mystery, hunting for a microbial culprit that is an absolute master of disguise. It's an imposter that's often mistaken for a much more famous pathogen. So let's get this investigation started. So that's the big question for us today, isn't it? I mean, picture this. You're in the lab, you've got an unknown sample, and all the first clues are pointing you straight toward a really notorious bad guy. But is it really them? Or something else hiding in plain sight? Let's find out. You know, every good investigation starts with gathering those first few pieces of evidence. We're right at the beginning of our case here, and we need to run some basic tests to just get our bearings and start narrowing down that long list of suspects. Okay, and here are our first two clues. So over on the left, the gram stain, it's showing us these little pink rod-shaped bacteria. That immediately tells us we are dealing with a gram-negative bacillus. Then on the right, we've grown it on McConkey agar. And look at that. It's popping with these vibrant pink colonies. Now, what that means is our mystery microbe can ferment lactose. So here's the key takeaway. We've got a lactose-fermenting gram-negative rod. That helps, but honestly, a lot of bacteria fit that description. And now we move on to the next test. And this is where our story takes a really deceptive twist. This one test is actually famous for causing confusion. It's like a classic trick question in microbiology. Yep, this is the big one. It's called triple sugar iron agar, or we just call it TSI. And it gives us a ton of information all at once. Basically, it's designed to see what sugars the bacteria can munch on and if it can produce something called hydrogen sulfide, or H2S. This is where our microbe's incredible disguise really starts to shine. And here it is. This is our result. Let's break this down real quick. You see that red slant over a yellow bottom? We call that K/A. But the thing that just jumps out at you is that deep black color at the bottom. That black stuff means our organism is H2S positive. Now here's what's so wild. This exact pattern, K/A with H2S production, is the classic textbook result for salmonella. So it's an open and shut case, right? I mean, it looks exactly like salmonella, a major pathogen we are always on the lookout for. Should we just call it and move on? Well, not so fast. A good investigator knows that when something seems just a little too perfect, you gotta dig deeper. And this is where we follow the full biochemical trail. We absolutely cannot rely on just one test, especially a tricky one like TSI. We need to run a whole panel of tests to uncover those subtle metabolic fingerprints that are going to reveal our microbe's true identity. Okay, let's add these to our case file. We see it's modal, which just means it can swim around. It's citrate positive, so it can use citrate as its only food source. It's MR positive, but VP negative, which tells us a little something about its fermentation pathway. And finally, it's urease negative. See, each one of these results is another piece of the puzzle. Now, there's one more key test that helps us separate two really close relatives in this family, the indole test. So our culprit, Citrobacter freundii, is indole negative, but its close cousin, Citrobacter cocceri, is indole positive. This is what we call the exam split because it's one of those tiny details that makes a huge difference in getting the right ID. So, we've gathered all the evidence, we've followed the clues, we've avoided the traps, and now it is time to put it all together. Let's make the final identification and unmask our microbial imposter once and for all. And there we have it, the complete profile of our culprit, Citrobacter freundii. It's a lactose fermenter. It gives that super tricky K/A H2S positive result on the TSI, and it has this very specific pattern of motility, citrate, and indole results. This is its definitive biochemical fingerprint. Now, let's see how our culprit holds up when we put it in a suspect lineup with some of the usuals. Look at the other common lactose fermenters here, like E. coli, Klebsiella, and Enterobacter. Now look all the way to that last column, H2S. Among this whole group, only Citrobacter is producing it. That's the thing that makes it stand out, but it's also the key to its salmonella disguise. And here it is, the solution to the entire mystery. The most critical clue was right there on one of our very first plates. Salmonella does not ferment lactose. It would have made pale, colorless colonies on that McConkey agar, not bright pink ones. So the moment we saw those pink colonies, we should have known it couldn't have been salmonella. Our microbe was an imposter all along. So
We cracked the case. But why does this actually matter so much? I mean, unmasking this little imposter isn't just a fun academic exercise. Getting the identification right has some really serious consequences for a patient's health.
This is the rap sheet for Citrobacter. Like other gram-negatives, its LPS can trigger sepsis, which is bad news. But what's really high yield, clinically speaking, is its connection to some very severe infections in newborns, specifically neonatal meningitis and even brain abscesses. This is not some harmless mimic. It's a dangerous pathogen in its own right, and doctors absolutely need to know what they're fighting. Ultimately, this whole case just goes to show you that in the world of microbiology, first impressions can be really deceiving, and making assumptions can be dangerous. Our investigation of Citrobacter freundii proves that digging just a little bit deeper is absolutely essential. And that leaves us with one final thought. What other microbial mimics are out there, and are you prepared to catch them?